So I inserted myself in the agenda because we did some work for Region 1 for rich fatness this last year. That's sort of a different take on the cyanotoxin effects in watersheds, so I thought I'd show it to you guys, just food for thought. And this is work Rich asked us to do, and basically these are all the people that sort of collaborated on it. Rosalina Christova at Cal State San Marcos cultured the algae that we were working with, and then Daniel Orr with Pete's group did the chemistry, as did Rafe Cadella at UC Santa Cruz, and then Jennifer and Bryn in our lab also helped with doing the toxicity testing data analysis. So I just wanted to acknowledge all the players. And I kind of wanted to just frame this in what we're doing at our lab related to algal toxins. The idea that we're expecting impacts for algal toxins to increase with climate change. So one thing we're working on, you guys all know it with SPOT, is we're sampling microcystin in sediments at all of our SPOT sites. And we've been doing this for, I think we have 3 years worth of data right now. And this is a trend monitoring study. And it's basically, you can think of it as analogous to stat monitoring. It's the same premise. These toxins, this particular toxin, binds to organic carbon in sediments. And so, and to other constituents in sediments. And so we figured we'd add this to SPOT at the base of our watersheds so that we can look at changes over time. And then more recently, we've also been doing bioaccumulation experiments, and we've demonstrated that microcystin bioaccumulates in one of our insect indicators in Chironomus. So we know that there's a potential now for trophic transfer of microcystin up into the higher end of the food chain. That's stuff we're basically doing for free, and we're going to pursue it. This is in collaboration with Aaron Stanfield at CSUMB.
Hey, Brian, what critter are you talking about?
Pardon me?
I missed the insect.
Chironomus dilutus. It's that little midge larvae, that little midge. So it's the fly larva. Hey, you have a question?
I was just curious. So you say it binds to sediment, microcystin binds to sediment, but you're not talking about biofilm growth. You're talking about dissolved phase microcystin binds?
It could be both, but it does bind. It's been shown to bind to clay in sediment and organic carbon in sediment.
Okay.
Yeah. But when we sample, we also could be accumulating some of those biofilms, with our samples. So for sure, we haven't differentiated between those two. So the thing that I want to talk about now are just neurotoxin effects. So with microcystin, it's a hepatotoxin. We don't really expect it to affect invertebrates, although there's some debate about that. But definitely with other algal toxins, some of them are neurotoxins. They act exactly like pesticides, and antitoxin is an example. And so Rich brought this to our attention this year because they had a couple sites on the Russian River where they had dog deaths. And he was curious about whether or not these algal samples could be affecting some of the invertebrate species that we work with for our surface water monitoring. And the extension is that this could be affect the same invertebrates that we're measuring in our bioassessment programs. That's what I'm going to talk about now. And so these were samples that were collected by Rich, algal mats collected by Rich, provided to Rosalina. She cultured them up. She isolated them. It's a fairly lengthy process to come up with isolates of these different species. And basically, the species is Phormidium. So she had 3 different strains of Phormidium that she cultured up in her laboratory, and then she provided those cultures to us. And so the idea was we were going to take the water that these animals these algae are being cultured in and see if they were toxic to our test organisms. And so this is what they look like. I'm sorry for the quality of this picture. It's just an iPhone picture that I snapped. It turned out it was the only picture I got, so I'm happy to use it. But it's 3 different strains of Phormidium growing in culture medium. The media is VG11 algal culture medium. And so we tested each one of these with Ceriodaphnia, dubia is just a pretest, and I think I showed you guys, I showed this at the symposium, that basically nuked everything in 2 days. So all the little daphnians were dead in 2 days. So they were highly toxic. So then the question was, we wanted to do LC50 work to establish toxicity thresholds and marry that to chemistry to see if the anatoxin concentrations are comparable to those that we measure in the field. And could there therefore be some effect on field organisms? So that was the idea. The first thing we had to do was use experiments just to demonstrate whether or not BG11 was toxic to our test organisms. So we did dose-response experiments with BG11. And we settled on these different ranges for our 3 test species, which are Hyalella, azteca, Chironomus dilutus, and Ceriodaphnia dubia. So the dose-response tests that I'm going to show the results of next are based on dilution series, like with Hyalella was 50% and down, and with Chironomus and C. dubia it was 25% and down. So when we're taking samples from those algal cultures, we diluted them by to 50% for Hyalella or to 25% for Chironomus and Ceriodaphnia. Does that all make sense? Okay. So then these are the test organisms. So it's Ceriodaphnia, which is representative of zooplankton, and then the middle one there is a midge larvae, Chironomus, and the one on the right is Hyalella. So those latter two are organisms that occur in our watersheds. Not necessarily the species, but definitely those genera. And you can think of Ceriodaphnia as a zooplankton. Then we did chemistry on these water samples, these dilution series. So we did ELISA for anatoxin, and then we confirmed this with LCMS-MS. So I'm going to show you those results as well. And again, the dilution series were based on those original BG11 tolerance tests. So these are the toxicity test results, and the y-axis are anatoxin concentrations given in microgram per liter based on the ELISA results. And so what I show there are the results of the 3 different strains, and these are either LC50 or IC25s or LC25s, depending on the species. And the basic take-home from this is all 3 of these strains were highly toxic to our test organisms. And if you look at the Ceriodaphnia results, there was about a half a part per billion based on total anatoxin concentration. I'm going to explain this in a second. The other thing to keep in mind here is that Chironomus test and that Hyalella test, those are just 96-hour exposures. So my guess is, especially for Chironomus, it's quite a bit more sensitive than those results indicate. And it's probably sensitive in the range of Ceriodaphnia, about the half part per billion range, if we did a longer-term exposure and looked at a more sensitive endpoint. So what we took from this is, wow, this stuff is pretty toxic. And it's not surprising because it basically acts the same as a neonicotinoid. It binds to the same neuroreceptor that imidacloprid binds to on the neurons of these organisms. So it's acting just like a pesticide in the environment. That's my guess. Okay, so does that all make sense? Okay. So just to reiterate, the culture water was highly toxic to all 3 of the organisms. The LCMS-MS results showed that no anatoxin A was present in these samples. And Rafe Goodell also measured saxitoxin and got no detects for those as well. So we're assuming that there's some anatoxin homolog that's present in these samples. I've had to go back and read about this a little bit because I don't really understand what that terminology means. It's the molecule's slightly different, right? Anatoxin A versus nanotoxin homolog. Okay. Do you know the answer to that?
Well, yeah. So there's some research that came out of New Zealand where they were looking at Phormidium mats in rivers, and they found that by far the bulk of the anatoxin was homoanatoxin in these mats, not regular old anatoxin.
Yeah.
So we might actually generally be looking for the wrong thing when we're doing this LCMS on straight up anatoxin.
Yeah. Okay. Yeah.
Do you know which ELISA kits were used by the lab to test for this?
Yeah, WPCL ran the ELISA test using.
Okay, thank you.
Just to be clear, the ELISA tests were positive?
Yeah. So that's what these histograms are, the ELISA results here.
Yeah, okay.
ELISA is cross-reactive across different toxin variants. And so you're going to pick all of them up with ELISA, whereas with LCMS, you're just going to pick up whatever your standard is for. And
Right. Right. But regardless, it looks like, so I don't really know if we can differentiate, like, the relative toxicity of anatoxin A versus homoanatoxin. Who knows?
So what are you measuring there with ELISA?
All of them.
Anatoxin. That's it.
Yeah.
Kind of like when you do a microsystem ELISA test, you're also getting non-genetic there. It's the group that you're testing for. It's the same person.
So it's appropriate to call it just anatoxin, or is it some other generic type thing? So we know, use the same language.
yeah.
Anyway.
So I just brought this up because I think we should be thinking about this now. Just add it to the list that we got of things that we got to be thinking about. Because
That's like.
because well, Rich, you can weigh in here now. But you know, my question is, what kind of concentrations are we measuring at these sites in the Russian River? And so there's definitely high enough concentrations that they're killing dogs. So that's why we went there in the first place. So what were the measured concentrations?
So during that year, most of the results we got in the water grass were low, under 1. There was when the dog died in Lower Russian, they actually went out 2 days later, the county health department did, and I believe they got a measurement, it was either 20 or 40 micrograms per liter in the water column. I forget which one it is, but it was the only time that we had that sample that high. This year we did a lot of work in the Russian. Most of our water graphs were non-detect. We had spats that were non-detect. However, I did species mat toxin analysis, and we had upwards of 8,000 to 17,000 micrograms per liter in the algal mats themselves of toxin. It's out there, but we weren't seeing it in the water for some reason. So the question is, did the dogs, were they getting it from the water when they died, or did they actually get a little bit of that algal mat and cause death?
Yeah, so, and I was also thinking about your results, which is why I cited your paper, you know, just the fact that we're detecting these things, benthic origin all over the state, but I was also thinking about the SMC study where you guys did Ceriodaphnia testing in Southern California watersheds and saw kind of the opposite of what we would expect, all this toxicity up in the mountain areas, but not down on the coast. You never figured that out, but based on the sensitivity of these guys to this type of toxin, it really makes you wonder. So that's sort of all I really wanted to talk about, other than, you know, the implications for surface water toxicity or surface water monitoring. We need to be thinking about this more as we proceed with our toxicity testing, and especially our bioassessment monitoring. So where we are right now is we're going to publish these results, and we need to be thinking about in-stream concentrations. At least these results give us a little bit of baseline information on how toxic these things are to the invertebrates. So when we measure it, I know everybody here cares more about, well, maybe not, but it's like human health and, you know, things like higher level organisms. But if we're doing all this bioassessment work, we should be thinking about this in terms of that as well, because there's a good potential for this to be another factor that's affecting our bugs. And then a question we have is just the relative sensitivity. You know, I actually look at Chironomus and Hyalella as native invertebrates, but there are also lots of other native invertebrates that we don't have information on that would be interesting to know how sensitive they are to these toxins as well. So I'll leave it at that.
And people should care anyway. It's the food web that is adversely affecting.
Yeah, I agree. But not just we're not just talking about drinking water.
No, I'm talking about anyone who wants to eat fish.
Yeah.
So this could be a cause of a low CSCI. It could contribute to a low CSCI.
Who knows? But
Perhaps something to investigate with a causal assessment.
Well, it's possible, but there are a lot of chemicals that have the potential to do that we don't look at either, right?
yeah.
There's this. I'm not at all dismissive of that. I think it's something to count, but
There were those chemicals that are widespread, perhaps.
I don't think we really know how widespread these are. We know that we have Betty's work. I would try to do that anywhere else. We're not. Obviously, I recall Betty from your stuff. We were looking at presence of the app producers.
Yeah.
But I mean, the antitoxin producers, but not necessarily the presence of the toxin.
No, we were also looking at this.
Yeah.
So we had hundreds of sites worth of data, actually it's only 1,200 or something at the time we published the paper. And a lot of those sites had species composition data just from the regular old swamp algae protocol. We asked crews, PSA, SMC, Region 2 did some sampling that they gave us. Basically take an extra filter of your homogenous when you do your rock scraping. Across your 11 transects. And then filter it the way you would have filtered for chlorophyll A, the same same prep steps that you would have used for filter A, and then we ground those up and analyzed them. And we analyzed all of the samples that ended up being a little over 350, I think, across the state for microcystins, and then a subset for some other toxins. We didn't get a lot of antitoxin A, but that's before we knew about the homoantitoxin. And so we actually got a very small, that was of all the of all the toxins we looked for and got some kind of a detection rate, it was the lowest for antitoxin A.
Like LTMS?
Yeah. Microcystins everywhere.
Well, 30% of your samples, right?
Yeah, practically everywhere. I mean, yeah, if we got toxin, period, chances are we at least got microcystins.
And that's the same number that Spot gets in their sediments. It's about 30%.
And USGS just started looking at wadeable streams. Same thing, about a third of their sites, they're getting microcystins.
What's interesting about Spot data is we see higher concentrations. I mean, it's not true of all of them, but surprising sites where you wouldn't expect it. Region 6 sites up in the mountain regions, we're finding it. So
yeah. For microcystins, by far the highest concentrations we got were in the Tahoe Basin and just in general sort of high elevation sites. But it was really gangbusters in Region 6 for some reason. And this is like in headwaters in some cases.
Same thing with Spot, so that's interesting. Well, it's not the same thing because we're not in headwaters, but
To add on about Region 6's observations, that's occurring in headwaters and higher elevations, it's actually a worldwide observation that researchers are reporting in the literature. They're finding it more and more in higher elevation waters that you would not expect having blooms and high nutrient inputs.
Are they finding it because they're looking for it? Or
perhaps that could be another thing, right? They're testing for it. Yeah.
Because people haven't been testing for this in headwater streams for very long.
Yeah, what's remarkable to me is how similar our results were to your results, including this big blank spot down here. Both studies found in Region 8, in the Santa Ana region, lower concentrations, right?
Yeah.
Didn't you find the same thing?
Well, I don't know if I would have identified Region 8 as lower concentrations. Actually, North Coast was probably the lowest. Again, just looking at wadeable streams. We weren't looking at large rivers, right? there seemed to be a dearth of hits up there, but everywhere else we were finding it. You know, there were hits distributed throughout the landscape. Again, for microsystems, that's where we got most of our hits.
So that's all I had to say. So 20 minutes, pretty good.
Hi, Brian.
Yeah.
I have one more thing to add about your question, about the different concentrations that were measured between the ELISA and the LCMS results.
Yeah.
I just looked up the ELISA kit that WPCL uses, and confirmed that for the different congeners of anatoxin, it has very high cross-reactivity with 2 other congeners, including homoanatoxin A. So that's perhaps one of the reasons why it was getting a detection there and not in the LCMS results, particularly since WPCL does not have a reference standard for homoanatoxin. So it is on the market, something that they could purchase in the future.
Okay, thanks for that information.
Yeah.
Another thought about thinking about this in the environment is that we don't really know if that the benthic algae that we are collecting and processing is available, right, to the organism that live in these environments, right? So you crush this stuff up and extract, release the cyanotoxin. So the question is that if for that to be affecting the community, it's got to be available. So
What Brian had done was they grew the culture in the medium and just drew up the medium. He didn't kill any of the...
oh no, I know, but that's
Can you guys speak louder, please?
Okay, so the question was,
sort of a highly concentrated culture, right? Or a monoculture of the
yeah, well, we did the culture for sure, but we diluted it down to, 1.25%, and that was still high.
So what's your recommendation?
Well, it ends up coming down to a forensic exercise, right? When you're trying to do your causal assessment, I'm just saying, don't ignore this. We're always limited by these analyte lists.
Yeah.
And so you just have to be thinking about other things that might be acting similarly. Like, for us, we do TIEs. And so the TIE, we go through all these processes to separate out what actually, what specific contaminant class is affecting the organism. So we would come to a point where we would say it's some organic contaminant that we can separate out with a C8 column, and we can bring it back and we do our chemistry on that for this whole suite of pesticides. If nothing's detected, then we got to start thinking about things that we're not measuring with our analysis. So that's what.
I was wondering if, Pete, anyone has looked at, in the PSA data where we've got both algae and bug data, has anyone looked at the presence of toxic species of algae and, the health of invertebrate? It might be interesting to see, are there certain species dropping out, for example, or, is there a correlation between poor scores and high and presence of toxic species?
This is Betty, I did a quick and dirty analysis trying to look at relationship between the toxin levels themselves in bugs, and I didn't really see anything major jumping out. As far as looking at species of toxin producers being there, in over 90% of our sites, they're there. So you're not getting you're not really getting two well-represented groups. I mean, they're almost everywhere.
Yeah, and that's kind of at the heart of where I'm coming from, is like, has this stuff always been everywhere, right? I mean, even in reference state, like before humans were here. Right? Well, go ahead.
Well, no, Pete brings up a really good thing, and when we were doing our study, that's one of the questions I wanted to try to answer with these benthic forms that grow in these wadable streams. You know, we have a lot of examples now in the literature of lentic water bodies, blooms getting worse, happening more often, spreading in geographic range, lasting longer, becoming more severe. But we don't have any baseline understanding about what's going on in wadable stream benthos. Nobody's been looking at it. They don't make these blooms that catch your eye, you know, pea green, right? So nobody's really been looking at it, and so we don't really have any clue whether the levels that we see right now are normal and business as usual, or whether there's something that's a mounting problem and we better be on top of it kind of thing. Like I said, some of our highest levels of toxicity or of toxin production were in headwater type, close to pristine type environments. And so that certainly doesn't lend itself to a belief that it's at least land use things that are contributing to it. However, atmospheric deposition, global warming, blah, 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 reduction in scour flows, I think that could actually be a pretty big player, and some of the analyses that we did in our paper pointed to that as something worth pursuing. We couldn't prove it, but it certainly was plausible. Phormidium does not form a really tight association with sediments, right? It could be wiped away pretty easily. So maybe if you have strong flushes in the spring in these headwater streams, you're not going to develop a big amount of biomass, of toxin-producing biomass. But maybe as we're not seeing as much snows in the Sierras and stuff like that, the paradigm could be shifting.
So your recommendation is follow up?
Well, I agree with what he said from the standpoint that I feel like the work that we've done so far, the knowledge that we've gained so far, means that especially if you're in a relatively unanthropogenically impacted area and you're getting toxicity, cyanotoxins should be the first thing you look at, I think. Now, if you're in an urbanized area, it might be just one of many things you look at. But I would say that if you're in one of these headwaters and you're getting toxicity, I would want to rule out cyanotoxins as fast as possible.
And it would be pretty straightforward. I mean, you can do it basically with some pretty minimal TIE procedures.
So we don't have any examples of toxicity in headwater streams attributed to cyanotoxins at this point.
Where the connection has been made.
Where the connections are made.
Conclusively.
The logical one would have been that SF. That would have been the logical place to look.
Yeah. And again, like I said, we found microcystins were very common. Other toxins, much less common. I realize now the anatoxin A, we're probably not looking really at it at the right way. So I don't even want to consider those results now that I know what I know. But microcystins all over the place. But, you know, are those hurting the benthic macroinvertebrates?
Yeah, and I don't think so just because of that. They're not.
Because of the mode of action.
Yeah.
But it might be worth looking into at some point anyway, because, anyway.
And what about incidents of dog deaths in headwater stream? I mean, that's another.
Yeah.
You know,
it would probably be harder for people to connect their dog death to a headwater.
Yeah, because they're like, we're in a beautiful area.
Where they cross the mountain stream. As opposed to a day of swimming and frolicking, right?
Yeah. And the water may be too cold for them to jump in.
Yeah.